Hi, everybody. Welcome to the Chapter 1 review. Keep in mind that these reviews are being recorded to help remind you guys of some of the major topics that we covered in these chapters. These reviews are not meant to cover every single thing that was covered in, Chapter 1 lecture, for example. So yeah, keep that in mind. Some of the stuff that appears in Chapter 1 might still appear on the exam. I just want to be sure that you guys are at least getting these major concepts down as sort of a refresher after watching that super long video. Hopefully, this will be a little bit useful for you guys whenever you're studying, as well as, just trying to understand some of the major concepts in the chapters. So let's just jump right on into it. So Chapter 1, we introduced ourselves to the concepts of what microbiology is and what we're trying to basically learn about in this class. So first of all, what are microorganisms? Microorganisms are organisms that are so small, they need a microscope to be seen. So micro literally meaning microscopic, right? So that's something that we would definitely help define this concept by. Moving on to cell theory. We talk about what we think of something being alive and how we might define something as being alive or being considered alive. One of the main concepts behind how to define this is cell theory. Now, that looks at things in the terms of cells, right? All living things are made of cells. Cells come from other cells originally. So a cell leads to a cell. They don't spontaneously generate. Cells have DNA as their genome. Cells have a membrane, and that's the minimum requirements for a living thing, right? So viruses can't qualify for this. And why is that? Well, we're going to learn more about viruses whenever we get in to Chapter 6 and get into details about them. But they're not made of cells. They don't have any of the features of typical cells. Some of them have a membrane. Not all of them have DNA. Most of them actually don't have what we could consider to be typical DNA. And when we learn about genetics and everything like that later on, and I guess that will be Chapter 9, I believe, then we'll learn a little bit about what it means to what a genome is and what DNA is and what its purpose is and how it functions for cells. And viruses, they do have genomes, but they basically can't do anything with them. They rely on the cells that they infect to use the machinery of those cells in order to propagate. So they don't qualify as alive based on cell theory. So that's why they aren't considered to be alive. Now, moving on to the prokaryotes. Prokaryotes lack a true nucleus. What is a nucleus? It's basically a membrane-bound structure in cells that contains the genetic material and structures associated with replicating that material and everything like that. Now, making copies of it, like when you're making new cells, you have to make a copy of your DNA, right? So prokaryotes, they can make copies of their DNA whenever they're dividing and everything, but they don't contain their genetic material or the structures associated with maintaining and replicating that inside of a nucleus. They have it in a region called the nucleoid region, but there's no membrane surrounding it to contain it. So that's the difference between prokaryotes and eukaryotes. Pro meaning before, karyo meaning like a true nucleus. That's what that term is referring to. So eukaryotes are, eu means true, and karyote meaning the nucleus, okay? So that's how we would divide those up. And it makes sense when you think about the structure of these organisms, that prokaryote that does not have a true nucleus or true organelles, which are other structures in the cell that perform functions in the cell that are membrane-bound structures. You may have heard of some of these, like the endoplasmic reticulum or the mitochondria. Prokaryotes don't have those. All of everything that goes on in a prokaryote just goes on within their cytoplasm, the goo that fills their insides, basically. So they don't have true membrane-bound structures inside of them, while the eukaryotes do. So that's the major feature distinction between those. So if we point out that prokaryotes don't have any of these kind of more complex structures associated with them, and then I go on to tell you guys that these were the first organisms on Earth, more likely than not, the prokaryotes. That makes sense, right? Because when we look at eukaryotes and they're more complex in their structure, the less complex stuff would more likely than not have come before the complex stuff. It gets a little bit more confusing than that, of course, whenever we start talking about the structures of the eukaryotes, the true membrane bound organelle containing true nucleus ones, whenever I start telling you guys that their membrane bound structures may have come from or evolved from eating up a prokaryote, and then that prokaryote became incorporated as a permanent fixture inside of that cell gradually over time. So that's a whole other concept that's called the endosymbiont theory, and we will talk about that more in another chapter. But in order to understand the big picture of all of this and how it sort of happened over time and how we're related and not related, then you need to kind of understand the intricacies of those. So, all right. So prokaryotes and eukaryotes make up all of the microorganisms on the planet. We can also include the viruses in there. They're non-living, right? So those are the microorganisms, quote unquote. All right, so most of Earth's atmosphere is produced through photosynthesis by microorganisms. So if I were to ask you guys a question on a test or a quiz, what are the #1 producers of, oxygen and eliminators of carbon dioxide, as we always think of plants doing, what organisms are the #1, producers of that? That would be microorganisms. And if we talk about specific microorganisms that would be associated with that, we'd be mostly talking about the algae. So those are our the protists, protista. But yeah, mostly the algae are involved in that. They have the same pigments that plants have, as well as some different ones as well. So not plants, okay, microorganisms. All right, pathogens, that's a term for any microorganism that causes disease. Some microorganisms can be harmless or even have a benefit to you having them as part of your body. Think about all the bacteria that are in your gut. Did you know that a lot of those guys are going to be breaking down foods that you can't break down so that you can utilize the nutrients that they've broken down? Bacteria do a lot of good things for us in our lives. They work to protect us. And there's a lot of bacteria in the environment that just don't affect us at all. Not everything is a pathogen. Not everything causes diseases. And yeah, there is gray area, right? We know that some people harbor, for example, Staphylococcus aureus or staph on their skin normally and naturally as part of their normal biome. And it's not harmful or anything. It just lives there naturally. But you introduce a cut, an abrasion, something like that, and that can lead to an infection. That could be considered an opportunistic infection. That infection wouldn't normally have existed if it hadn't been introduced to a deeper layer of the dermis, for example. Or it could be due to, a lowering of your immune system because you got sick with something else, or you went through chemotherapy, or you have HIV, or any number of things that can cause your immune system. Just stress can lower your immune system. And these things that might normally be associated with a healthy biome, a harmless biome, can now become pathogens as a result. So that's what we want to remember about pathogens. It's not black or white. You're not always a pathogen or, not a pathogen. Some of them are always going to be a pathogen, but then other ones just happen to fall into an area that they didn't belong, and now they can grow there, and it causes you, symptoms, bad symptoms, and that would be what we would be calling disease. Any deviation from the norm, in usually a harmful manner, is what we would call disease. So next is germ theory. So now we've recognized this concept of disease and disease-causing organisms, pathogens. Germ theory is the theory that communicable diseases that can be transferred from person to person, are caused by pathogenic organisms. And that's basically all that's talking about, that these organisms didn't spontaneously appear out of nothing. They didn't come from miasma. Back in the day, they thought that bad smells, just bad smell. If you smelled a bad smell, that smell was what made people sick. That is literally why plague doctors wore the masks that they wear. And they have all that, herbs and everything stuffed into those long, beak-like noses of the plague doctor masks to try to ward off the smells, because they believed that that was what was causing the diseases. Obviously, it wasn't, especially with the plague. We're talking about flea bites. Those are the vectors that are carrying the disease. Whereas the reservoir that was harboring the disease and keeping it alive in those communities would have been, rats and mice, rodents, essentially. Rodents would harbor the disease and be able to live with it and exist with it with few or no symptoms whatsoever. The fleas would bite them, And then whenever they go to bite the people, regurgitate some of those bacteria into the people, and then the people become sick with the plague, the black plague, the bubonic plague, the black plague being like septicemic plague. So that's like a whole system infection, specifically in the blood itself becoming infected with the bacteria. And then the pneumonic plague, which is in the lungs. Now, so these are communicable diseases, and you can't even get them from person to person. There are other diseases, of course, out there that are not associated with microbes, right? So cancer and things like that. We would not be concerned with germ theory as far as that goes. Now I'm going to throw you a curveball, which is to say some cancers are caused by microbes, like viruses. And one specifically that we can mention, of course, would be HPV, human papillomavirus, that causes cervical cancer. So that's an example there. So again, a lot of gray area here, but anytime that a disease is caused by a microorganism, those are called pathogens, right? And that when we do have communicable diseases, they have to be caused by something. They're not just caused by out of nowhere. You just spontaneously get sick from, thin air. So, but yeah, all right. Koch's postulates all right, let's just go to the scientific method really quick before we talk about Koch's postulates. Scientific method, we're using deductive reasoning to prove or disprove a hypothesis. So this is essentially a hypothesis is an educated guess to explain whatever you think might be happening. So deductive reasoning, we are going out and investigating what, aspects of that hypothesis to prove or disprove those parts of it, and then hopefully get to the answer of whether or not we think the hypothesis as a whole could be, proven or disproven by these results. So that's the idea of the scientific method using deductive reasoning. If we were talking about inductive reasoning, that's the sort of thing that like Sherlock Holmes would do, right? You don't have necessarily the end goal already in mind, that we are trying to formulate our own experiments to answer that question. Just picking up, information along the way is inductive. So, okay. So, Koch's postulates. This is a method that he used to identify pathogens. Basically, you take a sample from an organism that is ill with whatever disease we're talking about, and take that sample and culture it out and isolate organisms from it. Now, of course, if I take a swab of your throat, is that only going to contain the organism that is making you sick? Well, it's not. For those of you that might have said, yes, of course. No, it's not, by the way. How much bacteria is in your mouth right now? How much bacteria is in your throat right now? And you're not sick. So if I go and I swab your throat and I put it onto a plate, there's going to be all these different bacteria that are going to grow up. And that's normal bacteria. They're supposed to be there. They're helping out your body in different ways, like we talked about, even being protective in a way. But if we have overgrowth of some organism that could be new, introduced, or even just had an opportunity to overgrow the others and cause disease, then we get the disease associated with the illness associated with it, right? So I go in there and I swab your throat and I plate it out on some blood agar. That's for fastidious organisms, which means that these are organisms that would maybe require special growth factors, something that they can't make for themselves, in order to grow. We would use something like that in order to grow as much up as we can, and see what other organisms there could be. And then we can even go farther and separate them out through other methods like streak plates or dilutions, which we will talk about in chapter 3, and separate out the bacteria until we can get like cells. And a single cell will grow up into one colony. So when you look at a plate and you see one little bump there, that bump came from one cell that divided up and just, replicated itself. So we can use those colonies to separate out organisms. And Koch's postulates would take a sample from the, ill person or animal or whatever it was, and we would isolate it through that method of diluting it out, streaking it out on plates, all of that until we get a single microorganism. And then we can test the microorganisms that we see by putting them into another organism, like a, test rat or something like that, and seeing if they get sick with the same symptoms. And if they get sick with the same symptoms, it doesn't end there. We can re-isolate that same exact organism from this whole other animal that got sick with the same disease. That is what we would consider to be verifying the causative agent of that disease, right? So you re-isolated it and everything, got the same symptoms. That yellowish colony that grew on the plate made them all sick like this. But if we did the same thing with the white colonies that we grew, it was just normal throat bacteria. Nobody got sick from that. So that sort of stuff. That's how we isolate that out. And that's Koch's postulates as far as trying to determine causative agents of disease. Then we talk about in this chapter taxonomy, and this is just a way to organize all of these things based on specific traits that they may have. It could be based on their genetic relatedness or even just actual like observable features, whether or not they stain a certain color with a certain procedure or something like that. So in dividing things out based on taxonomy, we have domain, kingdom, phylum, class, order, family, genus, species. And the kingdom through the species, I'm sure you guys heard this before. We learned about this stuff in middle school and had to, know about which order was what then. You're going to have to know it again for this class, so do keep that in mind. That will be something that will be on the test. Domain is just allowing us to separate basically our prokaryotes from our eukaryotes. We used to just separate it, prokaryotes and eukaryotes, but our prokaryotes, the bacteria and the archaea, are our two groups that fall into prokaryotes. Archaea are they look like bacteria and they sort of behave like bacteria, but they have almost absolutely nothing in relation to the bacteria. They are prokaryotes, but they're actually genetically probably more related to the eukaryotes. And their cells sometimes have things more in common with the eukaryotes as well. They're not in any way related, really, when we break it down. They just looked like the similar thing. So we had them bunched into prokaryotes, but we wanted them separated more. So now we have 3 domains instead of just 2. And those 3 domains are bacteria and eukarya. And then those randos out in the wild, those are the archaea. The archaea, again, look like bacteria. They're microscopic like bacteria. They have similar shape. They live in a lot of the extreme environments. And we'll talk about those when we get into chapter 4, I believe. Yeah, that's prokaryotes. Okay. So that's taxonomy. That's just how we would be dividing everything up into their little areas so we can classify them and separate them out and recognize them more easily and identify them whenever we're doing tests to identify them. Finally, binomial nomenclature. So bi to nomial name. Nomenclature, how we name something. Okay, so binomial nomenclature, you have 2 names. Staphylococcus aureus, right? So, I mean, I don't feel like this is anything new or special for most of you guys, but that naming system, the binomial nomenclature of referring to a species, we write it out as the genus and then what we call the specific epithet. So the species name of something, like when we're talking about the bacteria that causes staph infections and MRSA, the genus that it belongs to is Staphylococcus. Staphylococcus has a lot of different species that can belong to it. Staphylococcus aureus or Staphylococcus epidermidis, and there's a whole bunch of others. So we can even break that down. The Streptococcus has a whole bunch of them associated with them. So that bacillus, Clostridium, a lot of these have a lot of stuff that belongs to that genus. So that's the first part of the name. The specific epithet is the part that helps us break it down even farther to the species, okay? So Staphylococcus is the genus. Aureus would be the specific epithet. That's just that second part of the name, okay? The genus is always capitalized. So we have that capitalized G there in the genus. The specific epithet is lowercase. So we have that lowercase s. So Staphylococcus, capital S, aureus, lowercase a, okay? And that whole thing, Staphylococcus aureus, is the species name when we're naming it using that binomial nomenclature, which we will be in this class. You also need to be italicizing it every time that you are typing it. Of course, when you're writing it by hand, just follow the capitalization and lowercase that we have going on here with the genus capitalized, specific epithet, lowercase, okay? These things can, cost you points if you're filling out stuff later on and you're not referring to things. This is one of the more commonly missed things whenever you are turning in your lab project with the unknown report and stuff like that. So do catch yourself on that sort of thing and try to use the proper nomenclature as well as just the whole system as far as naming them goes. So that's pretty much it for chapter 1 and all that we need to review for now. Again, after you've looked over this and get these major concepts down, if you haven't watched the full lecture already, I'm sure all of you have because you're such wonderful students. I would recommend going and watching that definitely before you go and take the exam so that you can go over more of the concepts that are covered. in that chapter other than just this one. This should definitely help you out with your quizzes and with your studying later on, you know, of course, after you've applied yourself as far as going over the whole chapter. I'm sure you did. So, all right, I will see you guys for the next review.